Welcome to the Cannabis Growth, Manufacturing, and Testing Learning Module Section 4, the Cannabis Extraction Process. A cannabis extract is a name given to any product which is made through the removal and processing of cannabinoids and terpenes from the source plant material. Cannabis extracts are typically much more concentrated than dried plant material, meaning patients can utilize less material for the same effect. However, the cost of processing cannabis extracts typically makes them more expensive than cannabis flower for patient consumption. Cannabis extracts can be used to make vape cartridges as well as many more traditional pharmaceutical dosage forms, such as pills, tinctures, topicals, or patches. Cannabis extracts also allow for the production of a final product with a known cannabinoid and terpene composition, which is appealing for practitioners and patients looking for a more traditional pharmaceutical dosing approach that can be challenging to achieve with the use of cannabis flower. There are two main methods of extracting the cannabinoids from the cannabis plant material. The first is solventless extraction. Final products of solventless extraction include dry sift, hash, and rosin. Solventless extraction uses mechanical techniques involving pressure, temperature, and filtration, and is typically more labor-intensive than solvent-based extraction. The second method of extracting cannabinoids is solvent-based extraction. This method uses chemical solvents to extract cannabinoids and terpenes from the plant. Products include butane hash oil, carbon dioxide oil, and distillate. There are three main methods of solvent-based extraction: ethanol, hydrocarbons, and supercritical carbon dioxide. Many extractions are labeled as solvent-free, but it is important to understand that these may actually be solvent-based extractions that have undergone a process known as purging, which evaporates away any remaining chemical solvent from the extract, as opposed to solventless extract, which does not use any chemical solvent at any stage of the production. Dry sift is a solventless cannabis extraction, which is a collection of refined trichomes that have been mechanically separated from the inflorescences. This typically occurs by agitating the cannabis over a series of fine mesh screens and results in a sand-like powdery resin. To make ice water or bubble hash, inflorescences are agitated in ice water, which is then filtered to collect the cannabis resin. The resin is then dried and pressed into hash. The final main product of solventless cannabis extraction is rosin. To make rosin, low heat and pressure are applied to keef, dry sift, or hash, which creates a resinous sap. Rosin is virtually indistinguishable from solvent-extracted oils, but has the advantage of being completely free of any solvent contamination. The first solvent-based cannabis extraction we will be discussing is ethanol extraction. This is the simplest solvent-based cannabis extraction form and involves saturating the plant material in ethanol, which then extracts the cannabinoids and terpenes from the plant material. The ethanol is then evaporated and recovered for reuse, leaving the extracted material behind. Ethanol-based cannabis extraction has less environmental impact than other forms of extraction, and ethanol is also non-toxic in low levels if any trace amounts remain in the extracted material. However, ethanol is an expensive solvent to purchase as large amounts are needed, and the resulting product is an impure crude extract. Ethanol is also highly flammable, so when used in a lab for extraction, controlled conditions must be in place to protect worker safety. The second main form of solvent-based extraction is through the use of hydrocarbons. Hydrocarbon extracts are commonly referred to as BHO, which stands for butane hash oil. Hydrocarbon extracts use pressurized chemical solvents like butane and propane to extract cannabinoids and terpenes from plant matter within a closed-loop system. Typically, dewaxing of the extract is built into closed-loop hydrocarbon extract systems. Open-loop systems also exist, but are incredibly dangerous due to the highly flammable nature of butane. In addition to the possibility of contamination if done improperly, hydrocarbon extracts have a high risk of combustion, and hydrocarbon extraction solvents are currently outlawed in Canada and banned in some U.S. counties due to this fact. The potency of the final product of hydrocarbon extraction is typically between 70 to 90%, with good preservation of both cannabinoids and terpenes. Hydrocarbon extract products can typically be used without further refinement, but the butane or propane must be removed in one of the following ways. The concentrate can be whipped over several hours to evaporate any remaining hydrocarbon and form a wax. Or, the concentrate can be spread into thin sheets and placed in a vacuum oven for at least 48 hours, which forms a product called shatter. Hydrocarbon extracts are popular because of the low startup cost to produce them. However, the flammability, expense, and loss of selectivity of the hydrocarbon process make it unpopular in large cannabis manufacturing facilities. The final main method of solvent-based cannabis extraction is known as supercritical CO2 extract. This method utilizes carbon dioxide under temperature and pressure to extract the cannabinoids and terpenoids from the plant. CO2, in its supercritical state, functions as both a gas and a liquid, which is an advantage in the extraction process. Using supercritical CO2, terpenes and cannabinoids can also be extracted separately as they extract from the plant material at different pressures. However, supercritical CO2 also extracts waxes and fatty acids from the plant material, which must then be removed through a process called winterization. Supercritical carbon dioxide extract is known as the gold standard of cannabis extraction, as it is very safe, has low environmental impact, and does not leave any solvent behind in the final product. However, supercritical CO2 has an extraordinarily high startup cost for the machinery involved, and the end result product must still be further refined in order to be utilized. As mentioned, supercritical CO2 extractions, as well as some hydrocarbon extractions, typically go through winterization and decarboxylation processes, and then may or may not be further distilled into pure distilled cannabis oil. Winterization is the process which removes fatty acids and waxes from the crude cannabis extract. The extract is dissolved in ethanol and then brought to between -40 to -80 degrees Celsius for 12 to 24 hours. This freezes the insoluble liquids and fats, which can then be removed through filtration. The ethanol is then evaporated away, leaving a refined cannabis extract behind. The extract is then heated to 100 to 140 degrees Celsius to decarboxylate cannabinoids from their acid form into their active decarboxylated form, which can be absorbed by the human body. Finally, the cannabis can then be exposed to heat and a vacuum to boil the cannabinoids, which then condense and collect in a more purified form. This final distillation step removes any remaining plant material, such as waxes and fats, resulting in a more concentrated, clear, light yellow oil, which can be used in a variety of medicinal formulations. Distillation also allows for precise control of concentration and chemical profiles, which is appealing to clinicians looking for a reproducible form of dosable medical cannabis.